So in this video, we're going to review the answers to some of the practice problems that we didn't get a chance to discuss in class. So the first question that we didn't get to review is question number 5. And for the purposes of making this a shorter video, I already wrote some notes on here. So the question is, how many different disaccharides of D-glucopyranose are possible? How many are non-reducing sugars? So to answer this question, let's start with, here's a molecule of glucose over here, and I've numbered the carbons. And this has a squiggle on the hydroxyl and the anomeric carbon because that can mean either it can be in the alpha or beta form. So the first thing we would do is we would figure out all possible combinations of monomers, and then all possible means of connecting the monomers. So in pink, you see the possible combination of monomers. We have alpha glucose and alpha glucose, or alpha and beta, or beta and beta, or beta and alpha. Now, alpha and beta and alpha are not the same because remember the first one that's listed, that's the one that has the anomeric carbon involved in the glycosidic bond. So these guys would have different glycosidic bonds in them. So once we have that in the purple, I've shown the possible ways of linking them. So you could link the two anomeric carbons, or you could link the anomeric carbon of 1 to carbon 2. You could also link it to carbon 3, carbon 4. You'll notice there's no alpha 1 to 5s here because 5 is the one that's used in the cyclization. So that does not have a hydroxyl available for glycosidic bond formation, but carbon 6 does. So you have 5 possibilities, okay, for each of the combinations of monomers. Okay. Now, what you'll notice is for the mixed dimers, I've designated the chirality at of the second unit when I was enumerating them, because that reveals to us that there is one duplicate. Okay, so if you have an alpha 1 to 1 and the second one is in a beta, and versus the beta 1 and the second one is in an alpha, these two are actually the same molecule. There's no way of distinguishing them. Okay, so we have 4 possible combinations of monomers, and then each of those has 5 possible ways of connecting them. So that makes 20, but we have one duplicate, so we have to subtract that out. So we have 19 possible combinations of the monomers. All right, so now let's draw, in the second question on this slide, gentabiose. Okay, so we've got the following information. Okay, it's a dimer of glucose, and we're told the glycosidic linkage is beta 1 to 6. And then on our second, the second unit of the disaccharide, it has its anomeric hydroxyl in the alpha configuration. So we'll start, okay, by drawing glucose, okay, and I already have glucose up here, right? So I'm going to start by copying that. And on the exam or the final, your reference sheet will have the structure of glucose. That's a terrible hexagon. There we go. All right, so this is my first unit in my glucose or my gentabiose disaccharide. All right, so the linkage is beta 1 to 6. So beta means that my anomeric hydroxyl is going to be above the plane of the ring. So there's my oxygen. Now, my second unit, okay, it's a 1 to 6. So I'm going to be linking up to this carbon on the second unit. So what I will do is I will draw that in. Whoops. So this is my carbon 6 right there. okay? And then I'm just going to draw, oopsie, I'm just going to try to draw a hexagon. There we go. So we draw another glucose unit in. And then we're told it's in the alpha configuration, so that means this hydroxyl is going down on that anomeric carbon. And so that is our gentabiose structure. All right, so now let's talk about the H antigens. So this is we talked about in class or in the previous video, blood typing. Okay, so the H antigen is the precursor to our A and B antigens. Okay, so the H antigen ends with beta D fucose, 1 to 2 beta D galactopyrannosyl, beta 1 to 3 beta D N acetylglucosamine. Oh my goodness, that's a lot. Okay, but we've got the structure of fucose here. We've got N-acetylglucosamine, and let's draw in a galactose. It's a C4 epimer of glucose. So remember that it is glucose. So I'll do galactose here. It's going to be glucose. All right, so there's my OH. There's my CH2OH. And we go, oops. With glucose, we go down, up, down. So I'm going to go down, up. At C4, now for galactose, I go up. Okay, so that is D-galactose. Okay, so let's draw. So the first unit is beta D fucose. So let's see, we'll put fucose in pink. Okay, so we're going to do beta D. So this is L-fucose. So D-fucose is going to have all of the chirality reversed, right? So we're going to have, here's my fucose. And so this is going to go down. This one would go up. This one goes up. And my CH3 is now going to be up. All right, so I've made my D. Now I need to make this beta. So beta is going to have that anomeric hydroxyl, same side as my 6 carbon. So that goes up, and it's going to have the oxygen for the glycosidic bond. And so here's my 1 of the 1 to 2. So now our next unit is going to be our galactose. We'll make that in purple. And so it's going to connect to the 2 carbon, which is this carbon right here. And that hydroxyl is below the ring. So I'm going to try to draw my ring so that I can so that I can reasonably, there we go. Okay, so there's my, that's below, so yeah, so there. And then the galactose, I'm going to draw in the remaining CH2OH. And all right, so that's my beta D galactopyrannosyl. And now I have a beta 1 to 3 linkage, so that's going to be up, just like the previous one. And then my N-acetylglucosamine, let's make that green, okay? All right, so my N-acetylglucosamine, I'm going to be linking to carbon 3. So we have 1, 2, 3. So that's going to be up. So this is going to be a little funky to draw, but that's okay. So I'm going to draw my hexagon here. And I have my OH down here. I have my CH2OH up here. And then I've got my N-acetyl group down here. All right, and so on carbon 3, it's up. So my linkage is, I would draw like that, so to indicate that it's, again, it's above the ring. Now, I was told it was a beta 1 to 3 linkage. This is my beta 1 to 3 linkage, right? And then I'm told it's beta D and acetylglucosamine, so I have to make this one in the beta form. And then my previous linkage was an alpha 1 to 2. Oh no, I'm sorry, it was a beta 1 to 2. Oops. Okay, so that is what the structure of the H antigen actually looks like in Haworth representations. Okay. All right, so let's look for our next practice problem. All right, so this is pectin, which is a polymer of D-galacturonic acid monomers connected via alpha 1 to 4 linkage. So if we wanted to draw a pectin tetrasaccharide, we would start with what does galacturonic acid look like. All right, and so galacturonic acid is a galactose. So let's start with our monomeric unit. So remember that, oops, hang on for a sec. Our galactose is, sorry, this one goes up, that goes up, that goes down. There's our CH2OH, and let's see. And then we've got, if it were in the alpha configuration. So this is alpha D galactose. So to make galacturonic acid, we replace carbon 6 with a carboxylic acid group for uronic acid. All right, so we're going to make 4 of those monomers. We're going to connect them via alpha 1 to 4 bonds. This is carbon 4, this is 1, okay? So I'm going to make my monomers, and I'm going to cheat a little bit by copying and pasting before I link them up. So copy, paste, paste, paste. Okay, so now let's do our linkages, and we'll do those in green. Okay, so we're doing alpha 1 to 4 linkages. So if you notice, the oxygen, the hydroxyl on the 4 is going to be up, right? So in all of these, I'm going to have the OH there, and then my alpha, okay, is going to be, my hydroxyl is going to be down. So my linkages are going to look like this. Oops, sorry. And then my end, the end, again, is not involved in a glycosidic bond, so I would write the little squiggle to indicate that that can open and close and undergo mutarotation. Okay? All right, so sometimes pectin contains branching. So let's add a branch point with an alpha 1 to 6 linkage at the second monomer. Okay, so now let's switch to purple for that. All right, so here's my second monomer right here. And I want to do an alpha 1 to 6 linkage. So I'm going to draw my hexagon. That's a very bad hexagon. I'm not good at this. This is why I don't teach organic chemistry. All right, so drawing in the remainder of my hydroxyls. Okay, so alpha 1 to 6, that means that I am going to have, that it's going to be between this carbon and my CH2OH here. So what I'm going to do is I'm going to erase this group here, okay, and I'm going to move this a little bit so that I can link them up better. There we go. All right, and so now I'm going to make my glycosidic bond between, that was not very good here. All right, my carbon 6 and the oxygen of the branch unit. So that is my branching, okay? So this here is what we would call the reducing end, because it's got a free anomeric carbon, right? And then these ends here would be called non-reducing ends, because they do not have free anomeric carbons. All right, so okay, so the next question, glycan groups of membrane glycoproteins and glycolipids. So which has higher structural complexity? A mixture of tripeptides generated from threonine, glutamic acid, and alanine, or a mixture of trisaccharides generated from xylose, glucose, and galactose. Okay, so right away, if we don't go into the numbers of it, we know, right, that because we have different ways of linking, that the trisaccharides are going to have more diversity, right? So more diversity because of different linkage types. And so, you know, it could be alpha versus beta. It could be 1 to different numbers, right? Different number of carbons. So without going into enumerating anything, we know automatically which one has more diversity, right? Because this is, you know, we have one way of hooking these up, right? So here we have threonine, glutamic acid, alanine, or we could switch the order of those, right? Or we could put the threonine last, and you know, ... We can only do that in 6 possible ways. So we've got 3 amino acids, and then times 2, and then we only have one way of linking them up. So that's 6 possible ones. Okay. In contrast, if we do the numbers out, we have 384 possibilities for trisaccharide. So remember, we have 6 possible linear sequences, just like here. Okay, so 6 linear sequences, just like the peptide. Right? But we have, again, we can have alpha or beta. Okay, so that gets us to 2. And then we have the different carbons that we could link up to. And then we have the possibility of branching instead of at that. And then, again, we have that second sugar that's linking up, and so that gives us another like 4 possible carbons it could be. So that winds up giving us 384 possibilities. Would I ever expect you to like go through this math like on an exam or anything? No. But you should be able to recognize quickly right away if you have two things, two polymers of the same length, and one's a peptide and one's a protein, or one's a nucleotide and the other is a carbohydrate. You should be able to figure out which one would have more diversity, and it's always going to be the carbohydrate. All right, pig feed can contain vegetables with high content of raffinose type oligosaccharides. And so the question is, explain why farmers premix this feed with alpha galactosidase before feeding the pigs to increase weight gain. Okay, so remember that raffinose has the alpha 1 to 4 bonds. All right, so alpha galactosidase, okay, cleaves or hydrolyzes alpha 1 to 4 bonds. And you can get that from the name, right? The alpha and then galactosidase, that means it breaks up that carbohydrate that is made up of galactose units, okay? So here's the deal, right? Pigs and humans, right, can't break alpha 1 to 4 bonds, okay? But if you treat the feed with alpha galactosidase, treating feed with alpha galactosidase breaks the raffinose into monomers. And so then the pig can use the monomers for energy directly. And they don't have to do any work to break that down. It's like it's almost like treating the feed with the enzyme is just going to feed pure sugar monomer units that can go directly into, glycolysis and those energy harvesting pathways. And so they can put on weight faster. All right, because again, it's been broken down into simple sugars. All right, so what causes flatulence in humans and pigs? As a reminder in class, right? It's intestinal bacteria have their own alpha galactosidase. So if you consume something that doesn't have alpha galactosidase in it, then what happens is the bacteria are going to grow, right? And so the products of bacterial, let's call it fermentation in your gut, are things like hydrogen and methane gas. Gases like hydrogen and methane. And so that's the thing that produces the flatulence when you consume those materials. All right, so let's look at, we go to the final practice problem. All All right, so this one we don't have to understand, the chemical method to be able to think about this problem here. So this has to do with branching, and so this is kind of the last two that we're going to do. All right, so if we want to know how much branching you have, so for example, the number, oopsie, wrong pen. The number of alpha 1 to 6 bonds, okay? This is how we would do it, okay? So we would treat a methylating agent, okay, that changes every hydroxyl to an OCH3, that methylates every hydroxyl. So all the glycosidic bonds are then hydrolyzed, and then you figure out how much of this 2,3-dimethyl glucose is there. Okay, so let's look at what a branch point would look like. So let me just quickly draw our glucose. Oops. There's my O. All right, CH2OH, and down, up, down. Okay, so that's my glucose, right? And if something is involved in a branch point, okay, what's going to happen is obviously my anomeric carbon is not going to be methylated, and this one's not going to be methylated because they're involved in the glycosidic bond. So what does that leave? Okay, well, notice that it's asking for 2,3-dimethyl glucose. So if this is at a branch point, what's going to happen is things are going to, the branch chain is going to grow from the 4-carbon end. So that will also be involved in glycosidic bond. So these three would be involved in glycosidic bonds, and they would not get methylated for the residue at the branch point. So only this one and this one. Okay, so that's why we're looking for the amount of the 2,3, because this is carbon 2 and this is carbon 3. Okay, so a branch residue during this procedure would become 2,3-dimethyl glucose. And ones that aren't branched would be 2, 3, and 6. 1 and 4 are also going to be, are still going to be involved in glycosidic bonds, right? Because that's how you get your chain, your straight chains. So you would have 2, 3, 6 di-O-methyl glucose. So that explains to you how we can figure out that amylopectin is less branched than glycogen, right? So we talked about how plants use amylopectin, humans use glycogen, and glycogen is a little more frequently branched. Well, this is the kind of experiment you would do to demonstrate that. Okay. So we're just going to skip this problem right here, figuring out percentages, because I just wanted to make the point of, like, this is how you would figure out how much branching there was. Okay, so the last practice problem we're going to do is kind of a case study in something called eye cell disease. So eye cell disease, okay, is a disease that is caused because you can't form a glycoconjugate properly, okay? And as usual, it's going to be the result of a missing enzyme, okay? And that missing enzyme, okay, is required to add a terminal mannose-6-phosphate residue, okay? So this is, let's see, here's our mannose 6-phosphate right here, okay? So in order to build the appropriate glycoconjugate, so here is where we attach the protein, right? And the rest of this is like the sugar that gets added on to the protein. And we know that, things like exactly which monomers are there and what configuration they're in are intimately related to their functional role. So if you don't have this mannose 6-phosphate residue there, it's not going to be able to do its job. Okay. So, and what happens is the purpose of this residue is to get things sorted into the appropriate position in the cell. Okay, so enzymes need to be sorted by the Golgi and to go into the lysosomes. Okay, so after the lysosomes have broken down macromolecules, contents will be released outside of the cell. And then, so receptors on the cell surface are going to bind the lysosomal enzymes, but they specifically recognize this M6P residue, the mannose 6-phosphate residue. And then what happens is they bring them into the cell. They endocytose them, okay? So what happens? I-cell patients have all sorts of negative impacts of missing this enzyme. It means they have developmental delays, short stature, they have skeletal abnormalities, and you really actually can't survive having this deficiency. And what you find is high levels of lysosomal enzymes are actually found in the blood, and that is what causes a lot of these problems. All right, so let's see if we can figure out, again, how I-cell disease affects lysosomal function. Okay, so I-cell disease. The missing enzyme means that lysosomal enzymes do not, let's see, so MC6P receptors bind lysosomal enzymes and bring them into the cell, right? So it means that the lysosomal enzymes don't get the M6P, so they don't wind up being correctly trafficked or correctly sent to the lysosomes, right? So if these enzymes that need to be tagged with this sugar right here, if they don't go into the lysosome, where are they going to go? Well, they wind up going and getting exported into the blood. Okay, so if they're not targeted to the lysosome, they get released into the blood. All right, so knowing what we know and knowing how this disease works, how could we potentially treat this disease? So one of the problems is a missing enzyme, right? So the key thing we would need to do, right, is somehow replace the missing enzyme. So maybe we could do this through gene therapy, right? For example, gene therapy. In this case, you give the person the gene, you introduce the gene for the missing enzyme, or you introduce a functional gene. Or you could have a purified form of the enzyme that if you could somehow deliver into the cell, right? So treat with a source of purified enzyme. So instead of them having to make it, you've got it as a biological drug that you can then introduce to them. But delivery is going to be an issue, in any of these cases. But so if the problem is a single enzyme, the solution is to provide the enzyme, okay? Because in this case, it's missing, not non-functional. So if it were non-functional, you might be able to find a way to use maybe CRISPR to edit the gene to make it functional. But here, it's just, it doesn't even get made at all. So, all right. Okay, so last thing. Researchers have identified a secreted glycoprotein enzyme, and they want to know if the glycan group is required for the normal function. So to do this, they make the glycoprotein in an in vitro system, which means they do it outside of cells, okay? And they purify the enzyme, and they do it with and without the attached glycan group, all right? So, in vitro assays show the enzyme actually works in both forms. So we have, so the key here is similar activity in both the glycosylated and non-glycosylated form. So they say that proves that the glycan group is not required, okay? So, give 3 reasons why this conclusion could be wrong, okay? So we've got similar activity, right, in an in vitro system. But what could the function of the glycan group be in cells that is not replicated in their in vitro system? So, perhaps the glycan group is required for getting the enzyme into a compartment, kind of like the lysosome one, right? I am losing the function of my pen. All right, so perhaps it's required for locating the enzyme in an organelle, okay? Or maybe the glycan group is required for exporting the enzyme. And maybe this is an enzyme that needs to be exported to do its job within the organism, okay? All right, so, and actually, it does happen to be a secreted glycoprotein, so there's that. And so in terms of locating it in an organelle, then maybe it's required to get inside a different cell type that would then target it into the cell. So then our last thing, right, would be that, glycan group is required for recognition by the target cell type that it's going to be. So we'll say in target cell type. So it could be, all 3 of these criticisms have to do with glycan groups are very good for having specific recognition, and so it's likely not to have anything to do with the enzymatic function, which was the only thing that was tested in the in vitro assay, and more having to do with targeting the enzyme and getting the enzyme where it needs to go. All right, so that's where we're going to stop.